Hello and welcome to my next video on the kidneys. Now, the kidneys are part of the renal system. Renal is kidneys. Here is a kidney. Now, the main parts is you have the capsule, which is on the outside of the kidney. Then you have the cortex, then the medulla, and then the pelvis. There are two blood vessels flowing into the kidneys. You have the renal artery, which takes blood to the kidneys, and the renal vein, which takes blood away from the kidneys. Inside each kidney, there are millions of nephrons. We'll look at what they do in a bit. Blood enters the kidneys through the renal artery and then passes through the capillaries in the cortex of the kidney. As the blood passes through the capillaries, substances are filtered out of the blood and into long tubules that surround the capillaries. This process is called ultrafiltration, which we'll look at in a bit. Useful substances are reabsorbed back into the blood from the tubules in the medulla. This is called selective reabsorption. And the remaining unwanted substances pass along the tubules, then along the ureter to the bladder, where they're expelled as urine. And then filtered blood passes out the kidneys through the renal vein. Now, this confusing diagram is a nephron. As I said, there can be 1 million nephrons in each kidney. Now, the important bit, it starts off in the top left. That's the kind of squiggly bit with a kind of up with a U. That is the glomerulus. The outside of the glomerulus is the Bowman's capsule. You then have the proximal convoluted tubule, which goes down. You then have the loop of Henle, which we'll look at in a bit. You have the descending limb, which is the one furthest to the left, then the ascending limb, which is one to the right. You then have the distal convoluted tubule before going into the collecting duct. Other important things, you have the renal artery feeds blood to the glomerulus. The afferent arteriole gives blood to the glomerulus. The efferent arteriole takes blood away from the glomerulus. You also have the renal vein, which takes blood away. Now, as you can see, it starts off in the cortex, goes down into the medulla, back out into the cortex, back down into the medulla. Now, the nephron, as we said, 1 million nephrons in each kidney, starts in the cortex with the glomerulus. The fluid moves away from the nephron. Now, this is where selective reabsorption occurs. Then goes into the medulla, where you have the loop of Henle. And the composition of the fluid changes throughout the nephron. Now I'm just going to explain that a little bit. Firstly, selective reabsorption is when useful substances are reabsorbed from the nephron into the bloodstream, while other excretory substances remain in the nephron. Now, the final product in the collecting duct is urine. Now this is, remember, the excretory stuff stays into the nephron and it will go to the bladder and then excreted. Now the fluid composition changes throughout. So in the proximal convoluted tubule, that's the one that's just left the glomerulus, the fluid is altered by the reabsorption of most sugars, salts and some water. In total, about 85% of the fluid is reabsorbed here. In the descending limb of the loop of Henle, the water potential of the fluid is decreased by the addition of salts and the removal of water. We'll look at that in a bit more detail. The ascending limb, the water potential is increased, and in the collecting duct, water potential is decreased and water is moved, and the final product, urine, has a low water potential. The cells of the proximal convoluted tubule have microvilli. These are very small and provide a large surface area for reabsorption. And this forms you have this from the filtrate in the tubules into the blood in the capillaries. So you, yet again, just to reiterate, you have glucose, amino acid, vitamins, salts, all reabsorbed by active transport and facilitated diffusion. They can't just diffuse. Some urea is also reabsorbed by diffusion. Water enters the blood by osmosis because of the water potential of the blood is lower than that of the filtrate and is reabsorbed from the loop of Henle and the distal convoluted tubule and the collecting duct. So the important thing, urine is made up of water and dissolved salts, urea and other substances such as hormones and excess vitamins. Urine shouldn't contain proteins and blood cells because they're too big to be filtered out, because remember the capillary only has small pores, so too big for any of the proteins or blood cells to leave, and it shouldn't contain glucose, amino acids and vitamins because they're actively reabsorbed back into the blood. If you have glucose or amino acids in the blood, particularly glucose, it could show that there is something wrong with blood glucose levels in your body. You could have too much glucose. You could have, diabetes. If there are protein or blood in your urine, then there's going to be a problem with the actual kidney itself, with the epithelial cells. There could be holes in it, dead cells. Not good. The glomerulus. It's again, all the kidneys is just really complicated drawings. Right, so you have the afferent arteriole goes into glomerulus, efferent leaves. Now, important thing to mention is that the efferent arteriole, the one leaving, is smaller in diameter than the afferent arteriole, so the blood in the glomerulus is under high pressure, which means it'll be squirted out, which is good. The high pressure forces liquid and small molecules in the blood out of the capillary and into the renal capsule or the Bowman's capsule. The liquid and small molecules pass through 3 layers to get to the renal capsule and enter the nephron tubules. You have the capillary wall of the blood. You then have a membrane called the basement membrane. In this case, we call them podocytes. We'll come on to what they are in a second. And then you have the epithelium of the Bowman's capsule. So this means large molecules like proteins and blood cells can't pass through and stay in the blood. And then you have the filtrate. Now, podocytes. These are epithelial cells of the Bowman's capsule called podocytes. They have very specialised shape. They're like little feet with little toes. We call these major processes. These ensure that there are gaps between the cells so that the fluid from the blood in the glomerulus can pass between the cells into the lumen of the Bowman's capsule. And then at the bottom of the glomerulus, you have the proximal convoluted tubule. Now, as we said, there is the endothelium of the capillaries, the basement membrane, and podocytes. And again, pressure differences in the glomerulus and Bowman's capsule means blood flows into the capsule. So, a bit more on selective reabsorption, as we said. The proximal convoluted tubule microvilli. They also have co-transporter proteins, sodium potassium pumps, and mitochondria. So, why do they have all these? Well, as with most things in A2 biology, you have sodium ions. Yay! So, what happens is this. You have co-transporter proteins. These transport glucose or amino acids in association with sodium ions. So, sodium ions will move out of the cell, and then they will be diffused back in using facilitated diffusion along with the co-transporter proteins, so glucose or amino acids enter the cell. Then, on the other side of the cell, you have glucose and amino acids diffusing into the blood. This reduces water potential in the cell, so water enters the cell and then into the blood, because you have a low water potential in the blood as well. Any large molecules, such as small proteins that may have entered the tubule, be reabsorbed by endocytosis. This is the loop of Henle. This is we found at the bottom bit we found in the medulla, and this is what controls the level of water. So water will generally go out of the loop of Henle, and others, like other molecules like sodium and chloride ions, will enter the cell. As you can see on the diagram, a single-lined arrow is diffusion, double-lined is osmosis, and triple-lined is active transport. The overall effect of the loop of Henle is to increase the concentration of salts in the tubule fluid, and then they diffuse out of the thin-walled ascending limb into the surrounding medulla tissue, giving the tissue fluid in the medulla a very low water potential, so water diffuses out. So, you have the filtrate flows into the tubule. Now, sodium and chloride ions diffuse into the descending limb, and water diffuses out. At the bottom, this means there is a low water potential in the tubule at the bottom of the descending limb. As you get the ascending limb, sodium and chloride ions diffuse out of the lower part of the ascending limb, and as you go up, they are actively transported out. This means that there is a high water potential in the tubule and a low water potential in the tissue fluid. Now, this means when you get to the collecting duct, all the H2O will diffuse out because there's a low water potential outside the duct. In a distal convoluted tubule, it is pretty much watertight, so no water can escape there. So any water that's in the tubule in the ascending limb will stay until the collecting duct. So this means water reabsorption occurs. Now, the loop of Henle is known as a hairpin countercurrent multiplier. This is how a countercurrent multiplier works. Near the top of the ascending limb, sodium and chloride ions are actively pumped out into the medulla. The ascending limb is impermeable to water, so the water stays inside the tubule, creating a low water potential in the medulla, and then because there's a high concentration of ions. Because of the low water potential in the medulla, then the descending limb, water moves out of the descending limb into the medulla by osmosis. This makes the filtrate more concentrated, because the ions can't diffuse out. The descending limb isn't permeable to them. Near the bottom of the ascending limb, kind of weird order of doing it in, but near the bottom of the ascending limb, sodium and chloride ions diffuse out into the medulla, furthering lower the water potential, and then these increase the ion concentration in the medulla, which lowers the water potential. This causes water to move out of the collecting duct by osmosis. So essentially, you've got the water potential being lowered in the tissue fluid at different areas at the same time, which means water is more likely to diffuse out. Now, in the collecting duct, as we said, high water potential, so water diffuses into tissues and tissue fluid, and the permeability of the walls changes the amount of water that is reabsorbed. This is called osmoregulation. Water is gained from food, drink, and during respiration, and water is lost in urine, sweat, water vapour in exhaled hair, and in faeces. Now, we need to make sure there's the correct amount of water in the cells and surrounding fluids, so we need to not lose all the water, and we also want to lose enough that we don't just kind of get too much. Now, depending on how much you drink and on the type of day, you'll produce either dilute or concentrated urine. Now, this is controlled by osmoregulation. The walls of the collecting duct have receptors which respond to the level of antidiuretic hormone, or ADH. ADH binds to osmoreceptors, causing a chain of enzyme-controlled reactions which insert vesicles into the plasma membrane, forming water channels. More ADH in the blood, more channels are formed. If less ADH is in the blood, the channels fold in and become vesicles. Now, the osmoreceptors are in the hypothalamus of the brain. If you remember, that's where quite a few hormones happen. Now, we're not 100% sure, but these cells probably respond to the effects of osmosis. When the water potential of the blood is low, the osmoreceptors lose water by osmosis. This causes them to shrink and stimulate neurosecretory cells in the hypothalamus. Now, these cells are specialised neurons that produce and release ADH. Now, the ADH is manufactured in the cell body of these cells, which lies in the hypothalamus. ADH flows down the axon to the terminal bulb in the posterior pituitary gland. It is stored there until needed. So, we just said that, just to clarify, water potential in the blood is monitored by osmoreceptors, responding to probably the effects of osmosis. Then ADH enters the blood and is transported around the body. ADH has a half-life of about 20 minutes, which means half of the concentration will go after 20 minutes. Now, this is a negative feedback loop, another one, yay. So you have an increase in water potential detected by osmoreceptors. Less ADH is released because increase in water potential means it is less negative, so there's more water. Then the collecting duct walls become less permeable because the vesicles fold in on themselves, less water is reabsorbed, and there's a decrease in water potential. If there's a decrease in water potential, it's detected by osmoreceptors, more ADH is released, the collecting duct walls become more permeable by forming more vesicle channels, more water is reabsorbed, and the water potential is increased. Kidney failure. Now, the kidneys can fail because of diabetes or hypertension, which, as you remember, is when you get too much glucose, or infection. Once the kidneys fail completely, the body is unable to remove excess water and certain waste products from the blood. This includes urea and excess salt. It's unable to regulate water potential. Other factors can also mean, if you have high blood pressure, this can damage the glomeruli, and otherwise the plural glomerulus, and then they can burst, and that'll cause many problems. So, waste products that kidneys would normally remove begin to build up. This can result in weight loss or vomiting. Fluid starts to accumulate in the tissues because the kidneys can't remove excess water from the blood. This causes part of the body to swell. The balance of ions in the body becomes unbalanced. The blood may become too acidic, and an imbalance of calcium phosphate can lead to brittle bones. Long-term kidney failure causes anemia, a lack of hemoglobin in the blood. So, what can you do? You can do dialysis. This is the most common treatment for kidney failure, and there's two main types. There is hemodialysis and peritoneal dialysis. In dialysis, removes waste, excess fluids and salt from the blood by passing it over a dialysis membrane. It is partially permeable, that allows the exchange of substances between the blood and dialysis fluid. This fluid contains the correct concentrations of salt, urea and water. Any substances in excess in the blood diffuse across the membrane into the dialysis fluid. And any substances that are in too low concentration diffuse into the blood from the dialysis fluid. Dialysis must be combined with carefully monitored diet. So the two, this one here is hemodialysis. So there's a machine that artificially does it. Now, a hairpin is added to avoid clotting. As you can see, the hairpin pump in the top left. This prevents clotting. A blood pump, that's pretty explanatory. A few different monitors, make sure everything's okay. Air trap and detector and an air detector clamp. So it's usually performed in clinics 3 times a week for several hours at each session, but some patients learn to carry it out at home. Now, as we just said, this is how dialysis occurs. So fluid flows over a partially permeable membrane. Hemodialysis has an artificial dialysis membrane. And peritoneal dialysis, the filter is in the body, and dialysis solution is poured in the space between the abdominal wall and the organs. The solution is then drained. Peritoneal dialysis is usually performed in several consecutive sessions daily at home or work. As the patient can walk around having dialysis, the method is sometimes called ambulatory peritoneal dialysis. Patients can feel increasingly unwell between dialysis sessions because waste products and fluid starts to build up in the blood. As we said, it can take 3 to 5 hours. It's quite expensive and pretty convenient, but it keeps people alive until a transplant can occur. Now, as we said, kidneys can be transplanted, and there are some advantages to having this and some disadvantages. Advantages: it's less time consuming, but you need immunosuppressants for the life of the kidney, because otherwise your own immune system could destroy the kidney, which means you're more prone to infection. Diet, less limited because you've got a new kidney, but it does require major surgery under a general anaesthetic. You'll feel better physically, but the risks of surgery of course include infection, bleeding and damage to surrounding organs. You have a general better quality of life. You can travel to other countries because you don't need to keep going to hospital, but you do need frequent checks for sign of organ rejection, but after a while that goes. And also it's just good to no longer see yourself as chronically ill. Side effects, of course, there is anti-rejection medicines cause fluid retention and high blood pressure. Immunosuppressants increase susceptibility to infections. Now, other important things that we can use urine for is testing for pregnancy, because pregnancy tests can detect human hormone chronic gonadotropin, HCG. It's only found in urine of pregnant women. Also, you can test for steroids, anabolic steroids and drugs that build up muscle tissue. Testosterone in an anabolic steroid, and there are other common ones such as nandrolone. Now, you can detect these in the urine as well. So that's all. And in conclusion, you have kidneys. There are millions of nephrons in there, and part of nephron is the loop of Henle, which is responsible for water reabsorption. You have osmoregulation, which is the control of how much water is reabsorbed. And if you have a problem with your kidney, this can lead to kidney failure, where you cannot work your kidney, and you'll end up getting many problems and will lead to death. So thank you for watching. As usual, any likes, comments, email me, the usual. And thank you for watching. This was actually my homework, so if my teacher is watching, I hope this is good enough. A, please. Thank you and goodbye.